In this video, I will break down gene expression and how genes result in unique proteins. Inherited traits are determined by genes, and the information encoded by genes is stored in a specific sequence of nucleotides in DNA. These are expressed through the synthesis of proteins and other RNA molecules involved in protein synthesis. Gene expression is the process by which DNA directs the synthesis of proteins and includes two major stages. Let's decode the process by which genes lead to the production of proteins. Transcription is the synthesis of RNA using information in DNA, where DNA strands serve as a template from which mRNA is constructed. Translation is the synthesis of a polypeptide using information in mRNA, which is translated into an amino acid sequence of polypeptides and ribosomes. So how is information in polypeptides organized? The answer is codons, triplets of nucleotides that specify a certain amino acid. Let's take a closer look at transcription. Messenger RNA is transcribed from the template strand of a gene. RNA polymerase is responsible for separating the two DNA strands and joining together RNA nucleotides complementary to the DNA template strand, elongating the RNA. Keep in mind RNA polymerases only function in a 5' to 3' direction, adding on to its 3' end. Transcription is split into initiation, elongation, and termination. In initiation, RNA polymerase binds to the promoter, the DNA sequence where transcription is initiated. The DNA strand begins to unwind, and polymerase initiates RNA synthesis at the start point on the template strand. The stretch of DNA that is ultimately transcribed into an RNA molecule is called a transcription unit, which begins at the promoter and extends several dozen nucleotides upstream. In eukaryotes, a transcription initiation complex is formed in order for transcription to begin. A collection of proteins called transcription factors attach to the promoter, allowing a binding spot for RNA polymerase II. Together, the transcription factors, RNA polymerase II, and in eukaryotes, the promoter DNA sequence called the tau box, form the transcription initiation complex. As polymerase moves downstream, unwinding the DNA and elongating the RNA transcript in a 5' to 3' direction, RNA transcript is released and the polymerase detaches from the DNA. The process occurs differently in bacteria and eukaryotes. In bacteria, RNA polymerase reaches a terminator sequence, causing RNA polymerase to detach from DNA and release the transcript, which is ready for translation. In eukaryotes, RNA polymerase II transcribes a polyadenylation signal sequence, which specifies a polyadenylation signal in the pre-mRNA. Once the signal is reached, it is bound by proteins in the nucleus, which cut the RNA transcript free from polymerase. The pre-mRNA then goes through processing before translation. During this phase of RNA processing, both ends of the primary transcript are altered. The 5' end receives a 5' cap, a modified form of the guanine nucleotide, while the 3' end receives a poly-A tail, or addition of 50 to 250 adenine nucleotides. These modifications are important because they facilitate the export of mature mRNA from the nucleus, protect the mRNA from degradation, and allow ribosomes to attach to the 5' end of the mRNA. RNA splicing also occurs during this stage, where introns are removed and exons remain and are reconnected. This is carried out by spliceosomes. Alternative RNA splicing, or removing different segments of a gene, can allow rise of different polypeptides from the same gene. Let's take a closer look at translation. Once the RNA is modified if necessary, the mRNA moves into the process of translation into a polypeptide. The start codon, AUG, signifies initiation of transcription. The translator, which reads the genetic message and builds a polypeptide accordingly, is called transfer RNA, or tRNA. tRNA transfers an amino acid from a pool in the cytoplasm to a growing polypeptide in a ribosome. Each tRNA molecule enables translation of a given mRNA codon into a certain amino acid, and the structure of tRNA allows it to do so. A tRNA bears a specific amino acid at one end of its structure, while at the other end is a nucleotide triplet that can base pair with its complementary codon. The protruding 3' end of the tRNA acts as a binding site for an amino acid, and the loop on the opposite end of the molecule includes the anticodon, which base pairs to a specific mRNA codon. The matching up of tRNA and amino acid is carried out by amino-acyl tRNA synthetases, whose active sites fit only a specific combination of amino acid and tRNA. Each ribosome has a binding site for mRNA and 3 binding sites for tRNA. The P site holds the tRNA carrying the growing polypeptide chain. The A site holds the tRNA carrying the next amino acid to be added to the chain, and the E site is where tRNAs are discharged. Once all amino acids for a polypeptide are added in the elongation process, the ribosome catalyzes the formation of a peptide bond, and a completed polypeptide is released through the exit tunnel. So what contributes to the final protein structure? Protein folding results in the three-dimensional protein with secondary and tertiary structure. Certain amino acids may be chemically modified by attachment of sugars, lipids, or phosphate groups. A signal peptide targets certain polypeptides to either the endomembrane system or for secretion. There are some exceptions that can cause malfunctions in this process. Mutations can result in a diversity of genes through natural selection. Signal nucleotide pair substitutions or insertions and deletions can have an effect on the final structure of a protein after translation and can be harmless or cause harmful effects on health or even diseases. Ultimately, genes are the codes that form proteins essential to cellular functions that keep us alive.